Okay, thanks for everyone coming. So I'd like to open up the opening of the second workshop on molecular communications. So we're hopefully running this as a yearly event. We ran last year's in Cambridge. It was very successful. So hopefully this one will be as successful, if not more. Hope you can see me in the darkness. It's just the way we can see this presentation. With regards to the Wi-Fi codes, we're just figuring out how these will be dealt out. So I know there's one code, they've said it's for everyone, but I think there's some issues, I think, with that. Today's program is generally going to be repeated for the next 3 days in format. So in the morning we'll have a keynote, we follow that by a tutorial, so primarily aimed at students and young researchers, but obviously anyone can attend from the sessions. We'll follow that with technical sessions, and then in the evening then we'll have a debate panel, and we'll run that same format for the 3 days. So hopefully it'll be a very interesting way to run a program, and very interactive. So what I'll do is I'll introduce the first keynote speaker we have here. Is Professor Tom Leenaerts from Belgium University. If you'd like to make your way up.
And thanks to the organizers for having me here. It's quite an interesting workshop, and I'm actually very looking forward to join you for the coming 3 days to see what's going on in this particular project. So as already told, my name is Tom Leenaerts. I'm affiliated with a number of universities. So the first one here is my main position is at the French University in Brussels. So for those not familiar with my great state of mind in Brussels, where everything is a bit surreal. We have everything divided in languages and all this kind of stuff. So I am at the French University as my main affiliation, but I'm also linked to the Flemish University partially. And in the meantime, we have built an bioinformatics institute which actually crosses both language communities and connects them back together. And today I will talk to you about one of my projects in computational biology, which I started as a postdoc. This is already 10 years ago. Because for me, I'm not sure what your backgrounds are, but for me, when you want to talk about molecular communication, when you really want to understand the biology, when you really want to go into the wet lab and the dirty pipetting of proteins, all this kind of stuff, as a computer scientist, you have to either decide to work in a lab where you have those things, or to dive into a molecular biology lab and to see what these people are doing, to understand what actually is going on. And so I did that for a 3-year postdoc to actually get acquainted with their language. So a lot of terms. So maybe raise your hand. Who has a biology background here? Very few. So so you will probably understand me better than the engineering, probably, because I'm going to talk about biology, molecular biology specifically. And so since it's a workshop about molecular communication, for me it was obvious to put on these slides, because I'm at the beginning, so probably other people are going to repeat this throughout this workshop, that in life there are different forms of communication, and the fundamental language of life is molecules, and there are different ways of communicating. Either the cell communicates with itself by secreting a molecule which binds them back to the original cell to induce some kind of state change, or you can have really cells being glued together and have some kind of channels allowing them to communicate and exchange materials, or you have the more known paracrine communication, which is used in signal transduction, where a cell secretes something and actually it reaches another cell in the vicinity that actually acts upon this, or the hormonal influences from endocrine communication, where through the bloodstream you actually get signals passing between different kinds of cells. And the cell is equipped to understand this language. Evolution has actually given it a way to deal with all these molecules floating around. And if you look at the molecular biology textbook, you most of the time get this kind of picture. Like a cell is like an empty white space with a few proteins in there. You have receptors in the membrane, and these receptors get information from the outside. By getting information from the outside, you have your first information transfer going on, which is a signal going from the outside to the inside. which requires some structural modifications in these membranes, which then bind to some other proteins and bind on and bind on and bind on until they reach the nucleus and express some kind of gene. Right? And then this gene goes back, it produces a protein, this protein has some action and so on. So this is very like a waterfall cascade-like of idea of how everything works in the cell. And people have been learning this in biology for a long time, and people modeling these things have been assuming that this works like that. They have talked about concentration, this concentration goes up, then it triggers other concentration and so on. And actually this is wrong. The point is that the cell is not like an empty space. Oh, sorry. It's not like an empty space. It's a very crowded space. It's a space where, like here, and not like here. So here you have a prokaryotic cell, and mostly in textbooks you see this, like you have this nice membrane, the flagella, and then in the middle you have this blue stuff, which is the DNA, and then everything is empty. And this is not how it works. This is how it works. This is actually what you have in a cell. It's a huge crowding of proteins that in some way have to get the job done by pushing against each other or by binding to something else and actually trying them, for instance, like to drive this motor, which drives this flagella to move the bacteria either in the direction of a source of food or either away from some kind of toxins. So keep this picture in mind. So this means that proteins are not just floating, diffusing around there, they're actually physically interacting. If you look inside, if you look at molecular biology and go see what those people are studying, they most of the time actually want to study these macromolecular complexes. So it's a combination of proteins that actually are glued or coming together, and together they perform this function. And sometimes you can replace one module by another one to actually change the function. So for instance, this is the TF8IIH protein, which is a transcriptome factor, but also transcription repair system, or DNA repair system. And so this complex of 7 different proteins actually is very important in expressing certain genes in your DNA. So it's not single protein that binds, it's a conglomerate, a macromolecular complex that actually does these kind of things. So it's like Lego, you could say. It's just taking the pieces, putting them together, and then I have my new thing. But okay, it's not really that simple, because evolution has selected for components that have a functional role to play. So I cannot just take any kind of block and build whatever that I want. There are certain rules that you have to follow in how these things are particularly constructed. And so the binding, if you then look again at biology textbooks and you think about how binding works, then most of the time you see this kind of pictures like this here. You see, for instance, you have this abstract representation of a protein, and this one has like a site which can bind something. For instance, you can have an effector or some ligand binding to this protein, or you can have another protein maybe phosphorylating, changing one of the amino acids residues inside the structure and modifying this protein, or you can other mechanisms. But it's a very fixed way of interacting. And it's also not that correct, but if you look at real proteins, they're more like this. They're a combination. One protein itself is a combination of different parts, and this part, for instance, here is the real functional site. So this part might, for instance, phosphorylate some kind of other protein, but you have other sites in these proteins that actually, for instance, are called the allosteric site and the substrate site. So the substrate site is, for instance, an element of your protein that first binds this protein in a particular location, and then it's in the right place to meet another protein to actually perform the functional effect on the other protein, like phosphorylation or something like that. But you have other sites, like for instance the allosteric site. If you bind something like this, it actually sends a signal through the structure and modifies this functional site. And so this is actually a very, very simple system that switches the behavior of this protein from binding this triangle to binding something else that's not a triangle, or simply to block the binding of that triangle. And this is actually done by this allosteric direct interaction. But it could also be that you have this allosteric site and it doesn't send a signal to this site, it simply inhibits the binding of this part of the protein to the area in the cell where it has to become active. So it can no longer bind this other protein and then activate the function. No, it actually is blocked because something is blocking this kind of activity. So what you see here is not simply like a fixed Lego structure that you put things together. Even you have to follow a certain plan because evolution says that they have to be functional units. But it's even worse, or even better, in my opinion. It's actually much more sophisticated. You have like little circuits that are proteins, and their structure allows you to manipulate how they function, when they are turned off, when they are turned on, how they have to activate in certain situations that will be active whenever they're not needed. So for instance, cancer is in many cases a mutation in, for instance, this kind of sites where the protein becomes consecutively active. It's no longer doing as it's told it's no longer operating like it was told to operate. And so one can study, for instance, now this idea of proteins having this kind of switching behavior. So you can say, okay, I have a protein B and a protein A binds and I have a communication through destruction, and I have a protein C that can bind. And you can use information theory to study under which concentration of A, B, and C do I get the optimal communication. And I've done that. It's a paper from 2008, but I will not focus on this story today. I can talk to you about this maybe after the talk or during the conference. What I want to focus on today is that the proteins in our bodies are not these simple blobs. The proteins in our bodies are even more sophisticated. They actually are composed of different kinds of modules. So this is one protein, a Src kinase, and this kinase has as a goal to phosphorylate certain proteins and the other proteins bind in this cavity, then it's phosphorylated, and then it's released. But in this state, the protein is inactive. The problem is that if it's in this state, this site is not open. And like I showed you with this schematic picture before, there's something binding, the tail in this case, to this module that actually blocks the opening of this active site. And as long as this site is not open, the protein cannot act. So in order to open this thing, you actually have to release this part, and then this part opens up and actually become active. So you have like a switch, like a toggle. Right? And you remove and you activate this protein by, here you see in the tail, you see a tyrosine, an amino acid tyrosine in the end, and a little P on top. This means it's phosphorylated. And when this tyrosine is phosphorylated, it binds to the binding cavity of this particular module. Yeah? So what you see here is a modular organization where you simply have to switch on and switch off this phosphor to actually inhibit or activate this particular kind of protein. Right? And by removing this phosphor, what actually happens is that the signal goes through this SH2 domain, and it tells the linker, you do not have to stay in this conformation. You can become flexible again. And by making it flexible again, this thing can open up and it actually activates. I'm giving a very simplified explanation how it works. But this is generally the idea. An interesting thing is that evolution came up with this kind of organization. So it actually came up with the idea that this particular active unit has to be combined with these two modules to actually produce this kind of switching on and off kind of behavior. So it's a modular organization like our programming that we do, like our engineering of circuits that we do. We have modular components that we put together, and we actually get some activity or some switching-like behavior in those modules. And biologists have already know this for a long time. They have been studying in the wet lab. They've taken this protein, this is the original sequence of the different domains that you have in this protein, and they simply start cutting them up and recombining them in different configurations. And what they saw was, okay, it really requires this configuration to actually have the proper switching. Otherwise, you actually lose this kind of behavior. So it's not randomly organized, these kind of things. Moreover, these blocks of SH2, SH3, and this kinase, so this part is the red part is kinase, is this part here, and SH3, SH2 are these two parts over here. You find them in many proteins. So these modules are reused all the time. Like in electronic design, like in programming, you use modular organization to actually recombine new parts to actually get novel functionalities. And this can be with the same modules, but this can also be different modules, like here, this fat module, this fur module, and you can imagine different kinds of combinations. Again, not all combinations are possible, because not all combinations are functional, but many of them are. And you also see, for instance, that this SH3, SH2 is very often appearing in the same combination. Yeah? And it's actually they're like a unit of two elements that actually is repeatedly used in many situations. So to summarize a bit what I maybe told you too quickly, so I'm sorry that I may be going too fast, but if you have questions, feel free. I just heard it has to be interactive, so please interrupt me. So you have, in molecular biology, these circuits, which we call these proteins, and these circuits are activated by upstream elements, and they activate themselves downstream elements, and they organize in different ways. And if you look at them, you can actually scale them from having a high integration, so it's one globular blob which actually performs all the function. You cannot identify any more modular effects anymore. And if you want to change the function, you have to really start changing things inside the binding site, but you see it's very integrated. And then you can go down this line to more and more modular organization, and more modular organization allows for more novel functionalities to evolve over time. Right? So people have, for instance, studied why are human beings more complex than other beings. In many cases, they have the same genes, they have the same proteins, so what is the big difference? The big difference is the wiring. because we have much more of these kind of things where we actually have all these modular components to actually link up with other systems. And you can actually look at these kind of things, and people in biology, a very famous guy, Wendell Lim, is a paper of him that I actually show here. This guy has been in California really like studying in the wet lab how to make these kind of circuits, how to make them functional, how to intervene in existing circuits and all this kind of stuff. It's very cool, and I always hope to have my own wet lab, but I've never been successful so far. But okay, this is very cool stuff. So as a computer scientist, if I want to contribute to this area, for me, okay, I would like to know where are the blueprints. Imagine now that I want to make my own circuits, and I want to design these communication channels myself, or maybe make novel communication channels with these kind of things. I need the blueprints. I need to have the wiring, and as in software engineering language, I want to have the design patterns for this kind of things. And so the story I want to talk to you about today is actually about unraveling this wiring in my attempt to interact with molecular biologists in finding how this actually works, to be able to predict how this works, and in the future maybe then design these kind of things. And so, first of all, okay, if these protein structures, if you bind something, then you have a signal going through and it changes something else, can you show me how this works? Can you show me the path and which amino acids in this structure are involved? So I need some kind of predictive method that actually allows me to analyze proteins and show this kind of information. It would be nice that if I can predict stuff, that I can find experimental data that supports whatever I predict about this structure, because then it confirms that what I'm doing is correct. It's just not random guessing. And if all these pathways I find them, can I show that they are functionally relevant, that actually have a role to play really in the protein? And like I showed you with this protein that activates and deactivates, with this SH3, SH2 and kinase domain. I mean, can I really show this pathway going from this ligand towards this linker, showing that I have that communication pathway? And since proteins are based on these modules, they have a lot of similar structural elements. Are they all functioning in the same way, or are there differences in their functioning? It will be like, is there only one circuit, or are there variations on a circuit? So that's the questions I'm going to try to answer in this thing. So we published some papers on that, and this work that I started in 2006 and then published in 2008 in this wet lab that I was talking about, and then I developed this further in my own group where we actually were able to link these to experiments. But let's begin from the start, how to identify those things. So in order to know where the wirings are inside this protein structure, I of course need protein structures. And since I want to understand how this wiring functions, if something binds or does not bind to this structure, I need two kinds of structure. I need a free form and I need a bound form of a protein. And if I take these two forms, I can then say, okay, this structure, so what you see are many of the same structures, they're more or less the same. So it cannot be that is the variation in the overall form of the protein that actually induces the communication. It has to be more subtle than that. And so the subtlety here is that amino acids have side chains. So the amino acids form this long gray backbone that I'm showing you here, but sticking out of this backbone are side chains. And these side chains define which kind of amino acids you actually have. And so what we were thinking was, okay, maybe it's the side chains, these things that are sticking out of the backbone are involved in communication. Because if normally they're flexible, but if I block this thing in a certain position, it might influence the behavior of neighboring side chains, and as a consequence, I might see some propagation of some signal going to this structure. So what we said was, okay, let's implement a software that takes a structure and then starts to move the side chains of this protein structure using Monte Carlo sampling. And I only allow, so this means I only allow movements in the side chains that are energetically allowed. Because a protein is in an energy well, and you don't want to have clashes between side chains, you want to have acceptable energetical conformations. So what you do is okay, I want to move this side chain, you calculate the energy state of your protein, and this thing will say, by changing the position of that side chain, you reduce the energy, so that's a good move. And you do this for all the side chains all the time until this sampling. And once in a while, you allow an error. You say, okay, because I want to sample the well of the energy, so once in a while, I will modify the side chain in a direction that is not energetically favored, that increases a bit the energy. And this allows me to sample this entire thing over the entire protein. And I can do this for bound and unbound states. And so, to show a bit more visually what I'm talking about here, and so this is in yellow is again this backbone, and I picked out one side chain here, and so I can move this side chain like I was talking about before, and you see this is since I have no limitations in my movement. Every conformation of this side chain has the same probability, right? But in reality, it's not the case. Not every conformation has the same probability. It could be, for instance, you have a neighboring side chain here, this one, that actually says, okay, I'm pointing in that direction, so you cannot move your side chain clashing with this one. So suddenly you shift your probability distribution from everything is equally probable to something that's more probable than something else. Right? And you have certain preferred orientations of the side chain given the neighbor. Right? And you can do this also with simple probability. You can also put conditional probability. So what is the probability of observing 2 if I know that this side chain has conformation 3? For instance, right? And then you can do this for all possible combinations for all the side chains in this structure, and this gives you probabilities, joint probabilities, conditional probabilities, and you already feel me coming, but because in the center you have the little plot, I can then calculate entropy, I can then calculate mutual information, and this mutual information allows me to tell, okay, the movement of this side chain, how much is this influenced by the movement of the other side chain? Or knowing the conformation of that side chain, does it tell me something about the conformation of the first one, right? And if I do this for all of them, go back. If I do this for all of them, and then I can actually have mutual information for the free form, mutual information for all the combination of the side chains of the bound form, and this gives me a way to look at the difference. So what changed? So if I look at the difference between the mutual information of the free and the bound form, I then can ask the question, okay, if I look at the difference between them, what has changed between the free and the bound form? And interestingly, all this blue stuff means nothing changed. But all the colored stuff means something has happened there. You have a conformational change induced by binding. And this is what I want to look at, because these are the ones that might be involved in sending the signal through the protein structure. So giving this matrix, I can then say, okay, look in this matrix with all these changes, because they're all possible changes I can find, and search for them those which are the most strongly affected. Yes?
Do you assume that the backbone always has the same structure?
Good question.
That the conformational change happened?
So good question. Here I use an ensemble of the same backbone. So I didn't go into detail there. So normally if you do protein studies, we can either study them by X-ray crystallography, and gives you one fixed structure of a protein, or you can do it via NMR, and then you have a structure with a lot of uncertainty about this. And so this is the ensemble of structures for this particular protein produced by NMR. So this gives me some slight variation in the backbone, and I take every backbone independently, and I do this sampling on every backbone independently. And I take all this data together. Good question, thank you. So if I have now this data, I simply use a clustering algorithm, and I use here the CAST clustering, and it finds for me the network of most influenced residues or amino acids inside the structure. So what you see here is a graph representation of my protein structure, and the links, so every node is an amino acid or a residue inside this protein structure. Every link is simply made between amino acids that actually are in proximity inside the structure. Okay? And so what you see here in red are is a network that actually I get from this data that is the most strongly affected by the binding event. And here at the bottom in orange is the interaction. This actually represents the peptide. So this represents this particular peptide binding to the structure. And it shows me that these guys here are the binding pocket residues, but you see that you actually have an effect going much further inside the structure. Okay? So you actually have identify in this way a communication channel that seems to be relevant for binding this peptide to this structure. So, hooray, we have a predictor, we find the wiring, let's move on. Is this actually true what we're looking at? Is this actually really what's going on inside this protein? And this was a very tough task, because it's a very tough question, and you need very fancy analysis techniques in biology to actually identify these things. But luckily I found an article by Andrew Lee and his students in 2004, another one in 2006, and they studied a particular protein which is called the PDZ domain. So this is the structure of this PDZ domain in its free form. This is the structure of this PDZ domain in its bound form, and here you see the ligand binding in this cavity between this beta strand and this helix inside this protein. So for me, this was interesting because they studied experimentally what residues or what amino acids inside this structure are actually influenced or dynamically affected by binding this peptide. Okay, so they had data, but this paper is from 2004, which means that the data is not available online in any database. So you have to go inside the paper and look like to very low resolution plots to see what actually is going on there in their paper. And so after communicating with these guys, it became clear that this plot was the most relevant one. This plot shows you based on what they call NMR metal group sidechain relaxation studies, which side chains are dynamically altering their behavior because of binding. So be careful. Before, in my prediction work, I looked at coupling between side chains. I looked at how one influences the other one and whether their movements are dependent on each other, yes or no. In this case, they just look at one side chain, which has a CH3 group on it, and they see did the rotation or dynamics of the CH3 group change, yes or no. But this was the thing that actually came the closest to what I needed as proof to see if my predictions made any sense, yes or no. And you see here the black peaks, so that the empty peaks were not good enough, the black peaks are actually relevant, and they show a number of them that actually are being affected. And they mapped this on the structure of this protein, and they said, okay, we found experimentally 3 regions. This red region, which is logical because it's directly the place where the peptide interacts with the protein, which makes a lot of sense. But they also saw some more distant ones, and they gave them the very original names, distal surface 1, distal surface 2. So for me, it's like, okay, this is very nice, because now if I have a structure of that protein and I do my predictions on that one, do I confirm what they actually have? They made a second paper, like I said, in 2006, where they said, okay, let's check if these things that we identify are functionally relevant. And they mutated a number of these residues inside this structure, and they observed that if they mutated residues on the areas that they predicted, they were more likely to see a dynamic effect than mutated residues which are not on the things that they predicted. Except for one, this I35, they did not see any results for that. But if they mutated this guy here, they also got a strong effect on the dynamics of this, on the functional behavior of this protein. So this was good. Then this means, this yellow bar, this is the experimental data. And then I saw that I was, of course, not alone. There is a very famous guy in molecular simulations called CARPLUS, and he does all these things with in force fields, manipulating these proteins at an atomic scale. And he developed a technique, and he made these predictions for this particular case. And then afterwards, there was 2 guys who had their own approach, Gerek and Oskan, who made a number of predictions. You see here at the experimental one at 14, this one had also 14, they had 26, so okay, let's see what happens. So my turn, let's see how this information theoretic approach actually behaves on this kind of work. And again, I made my prediction, and then I mapped my predictions on the structure, and very interestingly, this is the red area from before, this is the blue area from before, this is the yellow area from before, and I saw something more. I saw here in purple this I35 that I just mentioned, and I saw also in green another region that actually was affected. And so I was like, very interesting, but okay, I see a bit more than what's happening in the experiments. How well am I doing? And so I added my stuff here, and then I said, okay, maybe I should do some kind of accuracy analysis, like in machine learning. I should make a ROC curve, checking whether which predictions are good, which predictions are bad, and how are they actually comparing. And then finally, there was another approach, which is based on network analysis, on the entire hype of scale-free networks, where they have this between the centrality networks, and they claimed in the paper that simply looking at between the centrality gives you a very good prediction. You just don't need all those mumbo-jumbos of dynamics. This centrality is more than sufficient. And then we analyzed the predictive results on this PDZ domain, and so this line here is a random predictor. Here in green and in gray is between the centrality predictions. Here in purple was this CARPLUS prediction, and here in black is this Oskan and Gerek prediction. So these guys are just predicting random stuff. This guy is really random, these guys are also predicting random stuff, and luckily for us, our predictions are actually quite good. So what we see in this case is that by looking at the dynamics of the side chains and by analyzing their conformational coupling between those things, we actually get very good predictions of what's going on dynamically inside this protein. And if we apply this story, so we were also lucky because the one that we looked at was a human variant of this particular domain. We also had the structure of the mouse variant of this domain, and we analyzed also the mouse, and of course we had the same kind of predictions. And there were in yellow some slight variations, but they were easily explained by differences in the protein sequence or differences in the peptide that actually binds to this particular structure. So very nicely, we could actually map these things and we could show that whatever we predicted makes a lot of sense. And the results were great, but probably some people doing machine learning or data analysis in this room, confirming your results with one case is a bit weak. So this is the point that we should resolve. Another case was we saw this green area and that we actually predicted, but if you go back to the experimental data, this green area is actually here in the back. And there's a lot of data missing here and also here. There's a lot of data missing here in these graphs. So we could not confirm nor deny that this green area actually was relevant at all for this protein. So we said, okay, and then I took the jump. So I went from my safe harbor of computer science into the dirty area of molecular biology, and I said, let's make our own. And so I don't know how many people did that step, but for me it was a learning experience in the sense that you go to this molecular lab to say, okay, look, this is my results. They were very, very interested in the results. Let's try to now make our own, and let's take an example that is relevant. And so let's make our own protein structure. Let's look at the dynamics using this very fancy molecular dynamics technique. And then let's combine our results and see indeed whether we can again confirm what we're doing. And this was a project that took 6 years. So this is just to give you an idea of the work. Maybe if I would have hired a postdoc with all the expertise, and maybe would have gone faster, but we had to start with a PhD student to learn everything. So I mean, things took longer than anticipated. And we took a very interesting case, the case that actually I showed you at the beginning, this switch, this SARC protein that can activate and deactivate by releasing this particular phosphotyrosine here. If you dephosphorate it, then we said, okay, let's check indeed if we see communication from this side to that side and activating the protein, right? And so the only thing we had to do was produce structures for this one, do the metal relaxation experiments for this one, bound and unbound, with this tail, and then look at our results. And these results were recently published in Cell Structure. This is, like I said, a piece of 6 years of work. The interest in this protein was that it was also linked to diseases. In molecular biology, the first thing they ask you, which disease are you curing? It's like, no, I just want to understand the dynamics. No, if we do work for you, we need to take a protein that actually is relevant biomedically because this is important for our lab. And so FIN is very important for Alzheimer's disease. It was shown to actually be involved in becoming active in the case of people that have these beta oligomers that actually induce the disease. So we worked on the structures. And we actually, like I said, a long period of time, we found a novel structure of this ensemble of the FinSH2 domain. It was the first free ensemble that was available. We also made the first bound form of this ensemble with the tail of the original protein. This didn't exist before either. And then we said, okay, now let's look at the metal dynamics. So this side chain dynamics inside the protein, which is NMR. And all these blue balls are side chains inside the protein that have this CH3 group, which we can study in order to understand whether they're involved in communication. And I already put your number here. This is only 30% of the residues inside the structure. So we're looking at only a part. So the predictions looks at everything. So we will have to be able to join these two kind of data. And so we did it, and we managed, and the student was very happy that he learned all these techniques, and we found indeed that in this protein structure, there are certain ones that become more rigid when the peptide binds, there are other ones that become more flexible than the peptide binds, and here you see them on the structure. So you see most of the blue ones are becoming rigid, the red ones are some things that are becoming more flexible. So this is very nice, but it's only 30%. And then we said, okay, but on the one hand, we have now this experimental data. On the other hand, I have my predictor. I just need to find the best overlap between my predictor and experimental data to see which other ones, so next to this 30%, might be involved in actually signaling through this protein structure. And so I'm not going to explain this in detail. This fancy scheme explains how we did it. But in the end, we got a network. We got, again, this is here the peptide, and these are the residues inside the structure that are most strongly affected by binding this peptide to the structure, and we see again that we have the binding residues directly interacting with the peptide, but then we see also more distant effects. And we could map them again on the structure, and we can look on the front. So this is the front where the peptide is binding. This is the back, and you see that on the front, of course, of effect. But also here, this phenylalanine and this tyrosine here, which are very close to the linker, are actually also affected by this binding effect. And this is like 18 angstrom inside the structure that is actually happening. And so if we look at the top ranked residues and we map them again, so we see here some in the binding pocket, but we see a number of them, and these are very close to the SH2, SH3 linker, the one that actually has to get the signal like, now you can become flexible so you can activate the protein. And we can look at the data itself, but the most nice thing to confirm that indeed we had something relevant here was to say, okay, let's mutate these four. So if indeed they are relevant for inhibiting the function of the protein, if we mutate those four, then the protein will become active without actually toggling the switch, without actually activating the protein. And so I had to, I must admit, we first sent in the paper without this, and the reviewer said, nice paper, but we don't believe you, so find us proof. And we spent 1.5 years, so again, that's why it was 6 years, 1.5 years, first finding a group and then working with that group to produce those results. But in the end, we had results. In the end, this guy made a kinase inhibition assay, and this assay takes, for instance, this protein. It takes FIN or takes another protein. So this is the Src protein, which is the head member of this family, and FIN is a member of the Src protein family. And so here at the bottom, you see the increase in concentration of the protein CSK. And the protein CSK is responsible for dephosphorylating this tail. So remember that I showed that this tail is a tyrosine with a phosphor binding to the SH2. This protein removes this phosphor and activating normally this protein. Right? And so what you see here is that in the black line, what happens in a normal protein. So this is in Src, this is in FIM. You see that the more CSK you add, at a certain point, it activates the protein. It activates the protein. If you mutate the active site, then actually what you see is that this thing becomes active all the time. Oh, sorry, I'm actually explaining the wrong way. By adding CSK, it phosphorylates. I'm wrong. It phosphorylates the tail. By phosphorylating the tail, you decrease the activity. If you mutate the tail to something that cannot be phosphorylated, it will be active all the time. And it's the same thing here. If you add CSK, it will dephosphorylate the tail. It will deactivate the protein. And if the tail is mutated, it will remain active for a much longer time. So what we wanted to check was, okay, if we now mutate these four guys, will we be in this regime or in that regime? Right? And we want, of course, to see them in that kind of regime. And we made all these tests, and this is actually more or less a summary plot. What we saw was that mutating these three here, which are very close to the linker, it activates the protein without having to toggle the switch. This guy here, a bit more in the center, does not play such a sophisticated role. But the interesting part was that indeed we predicted that these were important for inhibiting the activity of the protein, and by mutating them, you're activating mutations of this particular kind of protein. So these results were again very nice. We were very happy. We published this recently, and this gives us a very good picture of how these things actually work. So how am I doing on time? How many? 10 minutes. Oh, okay. Oh, can go slower then. So this was very nice because we showed for the first time the role that this particular domain played in activating and inhibiting. This domain played in activating and inhibiting this entire protein. And since there are many members of this family of this particular kind of protein, actually our results are transferable to what actually is happening there, and people got very excited about that. So this brings me to the last question that I wanted to answer. Okay, this is now nice. But will every SH2 domain behave in the same way? Because SH2s are occurring in 130 different proteins, right? So I should, in principle, take all these 130 different SH2 domains and study their dynamics all at the same time and see then how are these circuits same or different. Right? Why do I want to do this? Because I cannot simply take any SH2 domain from another protein and plug it in here like I would do with a circuit, for instance. These components are, at a certain point, still evolved to fit the context of this larger protein structure. Okay? Sadly, we didn't have the time. Like I said, 6 years for one. And if I do this multiplied by 130, I'm a bit further in my life. So we were lucky for this particular domain. We actually found in the database of the proteins, UniProt, 8 different NMR structures, where we both had the bound and the unbound form. So this is the family that we had. So here, these are all the proteins where we could find these SH3s. And again, you see Fin and Sark there. The Fin is the one I'm studying here. So I had the SH3 domains of these guys bound to a peptide, but also from close family members. Lin and HCK are also Sark-related proteins, and they are very close. And then I had 4 which are very distantly related. So these are the structures. So if I zap to the next, so these are the 8 different structures. So these are Fin, Sark, Lin, and HCK, which are part of kinases with a similar format as the Sark kinase. And these are 4 other proteins that actually have also SH3 domains, but they have different other domains interacting with these particular kind of domains. And all of them had a free form and a bound form bound to a peptide, but never the same peptide. Right? There's always different peptides. But the case, in the end, we don't care. We're looking at what is similar, what is different between those things. So there can also be different bindings. So let's see what's going on. And we analyzed. We did not work experimentally here. We simply took the predictor. We analyzed all 8 structures, and then we said, okay, what are the results, and how can we compare them? And the simplest way to compare proteins is to take their amino acid sequence, to align their amino acid sequence, and then to see how similar and how different they are. And so here on the x-axis, we show the score of similarity between those sequences. The more similar they are to Sark, the closer they are to that particular protein, of course. And on this side, we put the similarity in the predicted positions inside the structure. Right? And so we plotted this, and we saw that proteins which are in sequence composition very similar to Sark, which are Fin, Lin, HCK, and these are much more different. They're also more similar in the way the communication channel looks like inside the structure, which is obvious, of course, but nobody showed this. Okay? And if we then do a correlation between all pairs, we very nicely find again, so this evolutionary tree shows the evolutionary relationship between the sequences. If we do this for the structures alone, we very nicely see that the darker, closer to red, the more correlation there is. The closer to blue, the less correlation there is. Very nicely see again this evolutionary relationship. So we see that Fin and Sark are very similar. We see that Lin, HCK are very similar, and that the group of Lin, HCK, and Fin and Sark are actually a kind of family, and that Bin, ASEP1, and the other 2 actually are very dissimilar from what's going on there, but they're also not similar among themselves. There are actually variations on this SH3 domain theme, which bind similar kind of motifs, but structurally and in terms of communication wiring, they're all differently organized than what you actually see there. Right? So this was quite exciting because this showed for us that, okay, there is a good explanation why you cannot simply do plug and play by taking an SH3, for instance, from this protein and plugging it in Lin. This will never work because it will not be functional. Yeah? So there's more there going on than only that. And we analyzed this thing in detail, and there's a paper is describing a lot of stuff, and we could actually annotate on-site SH3 structure, which things are typical for Sark-related proteins, which are typical for non-Sark-related proteins, what is common between all, and then we could look at this kind of things. And then we analyzed also what kind of amino acids, so is this always the same amino acid you find in that position, and you could actually see that, this plot. So if you look only at the Sark SH3 or of the model of all those that belong to the Sark family or all that do not belong to the family or all together, we always see the same signature. We always saw that those which we positionally predict, contain always more evolutionarily conserved amino acids than those that we do not predict. Right, so that's why always see the blue ones are those in the wiring that we predict. The orange ones are those that we do not predict. And if you look at the conservation of amino acids, we see that those that are predicted are always more conserved. And this allowed us to make a nice link to work of another famous guy, Ranganathan. He uses a sequence-based approach to what I do. I do it from a structure, he does it from a sequence. So in this case, he's not looking at how these structures are different. He's looking at what is the same between all those structures, what is the same in that family. And so, for instance, he analyzed also using sequences and statistics the SH3 domain, and he identified in what is common is like you have a number of them very important in the binding side, and then this kind of pipeline going through the structure. And if I look at my consensus model, if I take what is the same between all my structures, then again I see the same signature that we actually see in his data. But the last thing that we are convinced of is that, okay, it's nice to see, okay, what is common between the circuits, but it's also important what is different, because the differences might also indicate to differences in activity regulation. We'll quickly go back to this structure here. The Sark and Fin proteins are regulated by binding and releasing this particular kind of tail to this SH2 domain. But there's another protein called ABL, which for instance is involved in chronic myeloid leukemia, where the binding is not happening here. It's a completely different thing, but the structure of the protein is more or less similar, right? And so this is again composed of a kinase SH3, SH2, but the way the switch is organized, the way the switch functions, is different than from how the switch functions here. Right? And I'm convinced, and this is what I want to maybe show in the future, is that this switch that actually can identify this difference between those switches and see how to transport the switching behavior of the ABL to this protein and vice versa. But that's future stuff. And okay, we published this again in PLOS College Biology, and you can find all the details in that particular kind of paper. So I guess I was going a bit too fast. I was afraid I didn't have enough time, there are too many slides. So just what to remember, we can identify these long-range communication patterns. So we can identify the wiring inside these protein structures. We can do this by using information theory, and which information is a very practical tool to actually do these things. We predict in way better accuracy than the other techniques, which of course is always very nice if you do involve with machine learning. We actually, because these experimental methods are so difficult, this took us 2 years to get this dynamical data from NMR. If you have to do this for every protein in that way, you waste a lot of time. By having these computational approaches, you can actually do this very quite quickly. You only do structures, and they complement this experimental data. One thing I did not explain is that our data does not only correlate with the side chain dynamics of these NMR experiments. We have another technique which identifies changes in hydrogen bonds in protein structures, and also the result that we have here correlate with these changes in the hydrogen bonds in these protein structures. We showed also that the same protein in different species has the same kind of functional behavior, but also the wiring we can identify between different kinds of things, and the functionality is different aspects. It can change affinity, specificity, and stability of the protein. But there's still a lot to be done, and this is taking a lot of work, and now I'm working on my next project in this context, where I don't look at one single domain in the protein, but look at the combinations of the two domains. Again, this is a project of 4 years, and I hope in the end of the 4 years we finally will have the data to analyze that particular kind of combination. So now we're looking at this combination together. But the interesting thing is there is much data available. It's only a matter of finding people to work with and then getting the funding, I guess. So for instance, there's a friend of mine in Oxford who actually studies bromodomains, and he made an entire complete dictionary of all the crystal structures of bromodomains. And so we see there's many variations of those things, and they're very important in the context of epigenetics, the bromodomains. And so one of the things we would like to do is also understand how this works. And so, like I said, we need to scale up from simple single domains to domain combinations, because like I said, 80% of the proteins inside eukaryotic cells are multi-domain. So they're not one single structure, they're modular, they're flexible, and you want to understand how they work. And so the point is to actually look at more this kind of combination. So this work is done with many people, and I can also tell you a story about this here. So when I approached the NMR guy in Brussels to work on this project, his lab was not installed yet. So we were starting to do this kind of stuff, and they were still unpacking the magnets that were going to do the experiments, and they were not even tuned yet when we started this project. And now, finally, everything is installed and works properly. So these are people I worked with on this project. Elisa Cilia was a postdoc in my lab, Geerten Vuister, who is an NMR specialist now in the UK, people from Granada who worked on SH3s with me, then the entire group of Nico van Nuland, who I'm very grateful to him. Very sadly, he's become very ill and he had to stop his scientific career, but without him, I would never have been able to do this kind of research. And then my former postdoc lab where I started this line of research on domains. And thank you for your attention, and shoot, tell me your questions.
Thank you. So there's lots of crystal structures with and without the ligand. I was wondering if you looked to see if crystallographic factors correlate with your flexibility.
Initially, when I started that work in the lab, we first looked at crystal structures, which didn't work. And then we said, okay, maybe we can induce our own variation around the backbone without looking at the NMR data around the crystal structures. It didn't work, but this was in the beginning. So this was when we were first building that method, and then we found the NMR and then it worked. I would like to go back to crystal structures now. That's why I was pointing to these bromodomains, because there we have free n-bound of all these structures. But yeah, it's a question of money and resources at the moment to work on that question.
I can follow up briefly. So if you average over NMR structures, the variance of those structures corresponds very, very well to the B factor. So actually you'd expect it should work.
Yes, I know, but this was the confusing part. And then we focused on NMR and that worked properly. I mean, at that point, it's okay, we will go back at one point back to crystal structures. But yeah, okay, this was a slow, painful process. And now we can move back because now we know all those things and how they work. But okay, like I said, resources are in play here.
Sometimes
It works.
sometimes you can have one phosphorylation which changes the position of many domains.
Yes.
Will your approach will work in this case, for example, when not some residues, but the whole
Yes.
helices will move out?
Yeah, so this is large conformational changes, and this is what I'm not looking at the moment. So we look here. We looked first at simple domains because we knew beforehand that they did not have large conformational changes inside the structure. But you're completely right. This would be again the next step to actually say, okay, now we understand how the simple things work. Let's move up, and this was the scale that I was talking about. So now we look at domain combinations, how this works. And for instance, in this SH3, SH2 case, you can ask the question, okay, there's communication from the binding side to the linker, but does it cross the linker? Right? And people have looked at backbone dynamics, but didn't see anything, but nobody looked at sidechain dynamics. So again, we're trying to collect the data to see, is at the level of the sidechain dynamics anything again, and then we can later on combine this maybe with more larger conformational changes. And like in the kinase itself, there is also this kind of larger conformational changes going on.
May I ask? Yeah. So thanks for enlightening the audience on computational biology. I would like to ask from the engineering standpoint, in particular from the complex system design standpoint. So perhaps the question is naive, but would you please go back to our hypothesis slide?
So hypothesis slide.
You have one called back to our hypothesis.
Ah, okay. Yes.
So I have one very short reflection and two questions. The reflection, I was about to ask you about the connectedness of this network, but you already mentioned that it's close to scale-free network, right?
Yeah, but I mean, doesn't make any sense here because it's too small. I mean, scale-free only makes sense in large-scale networks.
It's a matter of scale.
So, I mean, for me, so people studied scale-free networks on large networks, and then they start doing scale-free networks on everything. And scale-free is something that you have to do on very large networks, in my opinion, not on this kind of
At
thing.
least in multiple domains where large.
Yeah.
Now, question number one would be, if you were about to transit or progress across different molecular archetypes, how would you define the cost function? Is it energy in each of the changes? How do you find energy in this context? Is it only chemical energy?
It's Gibbs-free energy. So the point is that the sampling is done in the context of a force field. Right? And we do the Monte Carlo sampling, and we allow only conformational changes of the sidechain conformation, if it's energetically allowed by the Gibbs-free energy function.
Yep.
And so it's in the context of a force field we do this, and then this gives us an idea of everyone's assuming the protein finds itself in this well where it's in the optimal state, but we sample then the sidechain configuration inside that well.
Yeah, interesting. Now, that triggers the question, is it only energy that matters? Can you think about at large resources? Do you need molecules, the structure, molecules to build the different blocks? Do you need molecules to communicate among different structures? Is it only that type of energy that matters, or resources at large?
So really designing that's a good question. I would not immediately have an answer for that. I have to think about that. But I'm pretty much convinced that you can design these proteins. So giving the information that I have here, I can come up with a sequence of amino acids that actually gives you the same functional behavior as what I see in real proteins. And this actually, you can actually design those things. But the problem that you have there is that proteins are promiscuous. They bind promiscuously. And so an engineer does not like promiscuity because he wants to have like, this is my target, you bind to that, you only have that functional behavior. And this is something that nature did not do. And I mean, that will be more difficult to design, in my opinion.
Very last question, can you show a slide called consistency in families?
Consistency?
In families.
This one, yes.
So again, from the abstraction of complex system design, how could you capture the different attributes of each of these archetypes or entities? How could you quantify qualitative attributes like robustness versus flexibility? These are typical qualities in design. Does this make sense in this context? What do you think about it?
But this makes all sense because you have there's an entire field in biology called protein design. And in that area, they talk about robustness and flexibility. The protein is not a static structure. And I hope that I made this clear by I look at these different kinds of backbones. The protein is a thing that actually is flexible. As soon as it becomes rigid, it cannot function properly anymore because you need more energy to bind peptides and things like that. It's the flexibility that actually allows you for this. So the people have developed theories about this and say, okay, a protein is flexible and it actually visits every possible conformational state, even the bound. But it's not staying in that state, it's only occurs with a very low probability. By binding, you shift the probabilities of seeing a certain state towards the bound conformation. And so by all these allosteric effects and dynamic effects, you shift the proteins to certain areas that allow it to accept certain binding partners or to reject certain binding partners. And it's this kind of dynamical process that actually allows you to describe this functional behavior. And this has to be flexible, this has to be robust, that if a mutation happens there, then you're still going to go through that pathway. I mean, nature designed it in that way to actually be like that.
If there's no further questions, I'd like to thank Mom again for the...
Thank you for your attention.
Just some information on the program for the rest of the day. Very interesting.
Thank you.
So you'll have information on the registration booklets there as well. So the rest of the day would be, we have a coffee break now until 11 o'clock, followed by a tutorial for 2 hours, and this is going to be run by Gianluca Reali, looking at the molecular communications toolkit and the language. And that would be basically tools developed from the CERPA project, which is funding this event. After lunch then, we have going to the technical sessions. And following a short break then, we will end by today with a debate panel on looking at the challenges of young researchers entering the domain of molecular communications. So thanks a lot, and we'll have coffee outside now. Thank you.
Vogliamo il suo Apple device, maybe it doesn't.
So io.
Pensavo fosse altro.
Lo stacco? Faccio io, sì. Un po' deformato. Aspetti, scusi, ma faccio staccare. Non è una bella cosa. Un po' deformato il connettore. Ok.
So you can use for large factor. As soon as you go to smaller factor, you want to understand the detail, then you that's how I see it in my work.
Siamo salvi? Siamo salvi.
And in the end, as a shrink paper, you have to find a way to doing the rest. Yes, yes, yes.
Problema del computer loro, questo sta facendo. Questo gli altri presentavano, c'era un bel margine dal soffitto.
The first was really like they can be identified as dynamical aspects and then understand better how things work, maybe one design things. Like I said, I was a little bit like myself, so it's a bit hard to come through. So getting an scan paper was on a level and I showed you that
Sta facendo lei? No, perché ci vuole questo. Ma tanto ce l'ho io. Non lo lascerei qua però.
Terra? Dentro? Vuoi chiudere e mettere dentro?
Se non spostiamo un po' più in qua. Ah, sicuro, no? Ah, sì.
Professor Real, you next week tutorial on the molecular ML.
But for me was most, I didn't go back to crystal structure simply because I don't have the experimental proof what I was seeing made sense. And then at that point I found NMR and said, okay, let's first take that road, and because there I know there's data. I actually can I refuse whatever I'm doing right now.
But there are some freaks now.
Now it makes it So this is nice thing. The PZ case has the crystal structures. So it has the crystal bound and unbound. And so the in between us network is based on crystal structures. Not on doing the sampling. So I would still like to go back to the sampling of the side chains of those guys and see how this happens. But now we also another guy actually was looking at conditioning, so see how our results, and now I look at the sidechain confirmations, and I don't take the backbone into account as in my calculation. And it seems that what I saw now, actually a guy was one here in my group looking at this, we saw that if you, as soon as you take your movement relative to your backbone is no longer working anymore. So I want to understand why you see it in one case and not in another case. This is work there. A lot of things to be done. The last year. I have to talk anyway when we're going to discuss it.
Approach, the logic information changes can be treated just like one mutation, for example, can influence a cluster.
But I think it will be the connection that changes really. So there you really have two networks, one in one confirmation state and another confirmation state. Yes, yes. So then again the question on the data would be like, would you need intermediate steps, or what time scale you're working? Because the side chains are working on nano time scale, milliseconds time scale, and so the larger chain would be like slower. So it would mean that you have to combine time scales in this model, and I'm not sure how to do that yet. This is something that would be puzzling to me, because then you have two networks which are actually at different scales of time. It would be interesting to me.
What I'm asking, what I'm trying to do now is to make just kinetical model, not only a model of but then we just know the date of a couple of important movements, and we just try to treat it just like a physical-chemical transitions and apply all the rules like the conservation equaling the free energy should be zero amongst all the just to try to see when, because there are drugs which target the kinase in different conformations.
Yes.
How can they work together? When one drug targets one conformation, another not.
Yes.
But these are all wide changes when all the helices is moving and so on. So
There was this lady, what's her name, and she actually also studied this back, so in the kinase you have a kind of backbone,
and she studied the effect of the backbone. I forgot her name.
Spine. They call it spine.
The spine, yeah, but this lady actually found it. I forgot her name. Yeah, I saw her.
From where?
States, I think. She's from the States. Yeah, I forgot her name. But she has some nice work on molecular dynamics models also of the spine and how the spine changes and when activating the kinase also. For me, it would be also a nice step to say, okay, let's take the kinase in these different configurations and then do the same analysis and to see how we can compare those endpoints.
But actually it's very interesting that you don't need crystal structure, because for some of the proteins there are no crystal structure of the co-protein, just like kinase domain and so on. Here you can modulate. But are you sure that you can properly calculate the entropy during calculating of free energy? Because entropy is something that you can calculate different states to see how it's flexible and so on.
You mean the entropy of the side chains? Yes, indeed you can, and that's what we do. We move them, but I think what we do is, what is that we have an alphabet, we have a confirmation alphabet for every side chain. We simply look at probability of observing certain confirmational states in that alphabet. And this gives us a huge, for every position, a huge number of, for every residue, a huge number of positions that you can find these confirmations, and this we correlate with the huge information. And this gives us a way to look at confirmational coupling between side chains. But you could probably check also entropy and see the entropy changes and see how it relates, and it will be closer to what we see experimentally.
So we can use the
But like I said, this is an ongoing project and not many resources.